This is Chapter 8, Part B, Microbial Genetics. So remember from Chapter 5, we said bacterial cells carry out an enormous number of metabolic reactions. But the common underlying feature in all of these metabolic reactions is that they utilize enzymes. Remember, enzymes are just proteins, so they're going to be synthesized like every other protein via transcription and translation. So in this section, we'll look at some mechanisms to prevent synthesis of unwanted enzymes. Many genes, roughly 60 to 80%, are not regulated and instead are considered constitutive. So these constitutive genes are constantly expressed at a fixed rate. So ultimately, they're always turned on. So you can think of them as the genes that help to constitute or make up an organism or a cell and its function. So these genes that are turned on all the time typically are going to code for some of those housekeeping genes, regulatory genes. So enzymes that the cell needs in large amounts or needs for those major life processes like glycolysis, right, in their metabolism. So protein synthesis requires a huge amount of energy. Cells want to conserve energy by only making the proteins that are needed at a particular time. Right? So ultimately, genes are only expressed as needed. Right? So we don't need to express all of our genes in our DNA at the same time. Some genes are expressed while others are not. So your inducible genes are normally turned off, so we only turn them on as needed. Repressible genes are normally on and must be turned off as needed. Right? But ultimately, both of these categories are our regulated types of genes. So we can either turn them off or turn them on. So we can regulate our gene expression during transcription, right? So transcriptional control, or during translation, right? Translational control. We can also have post-translational control. So after we've made the enzyme in the protein, maybe we can still inactivate it with maybe something like one of those competitive or non-competitive inhibitors. So repression is going to repress or inhibit our gene expression and ultimately decrease enzyme synthesis. So this typically is mediated by regulatory proteins called repressors that are going to physically block that promoter binding site for the RNA polymerase to undergo transcription. So normally, without any repressor, our promoter, remember that start region for transcription, is open, so RNA polymerase can bind to it and transcription begins. But a repressor protein is, like I said, physically blocking RNA polymerase from attaching to that promoter. So if RNA polymerase can never bind to the gene to transcribe it and copy it, then we won't be able to synthesize that protein. The default position for repressible genes is on. So normally they would just be on. So we have to repress them and turn them off, right, as needed. Induction is going to turn on gene expression. So it's initiated by an inducer. So think of when you induce labor, right, you're triggering labor to begin. So we're triggering gene expression to begin with induction. So these inducer molecules are going to turn on this gene expression and make available that promoter so RNA polymerase can bind and transcribe. The default position for inducible genes is off, right? So we can only induce or turn them on as needed. So any other time they would be off. So in the Operon model of gene expression, the promoter is still that segment of DNA where the RNA polymerase is going to initiate transcription of our structural genes. The operator is the following segment of DNA that's going to control transcription of these genes. So the operon is kind of the whole thing together with the operator, the promoter, and the structural genes that they control and regulate. So a common example of this operon model of gene expression is the inducible lac operon or the lactose operon. So again, in these inducible operons, these inducible genes, they're not going to be transcribed unless that inducer is present. So unless that specific OK signal is given, transcription is not going to occur. So in the lac Operon example, so this cell produces enzymes for lactose utilization, right, to digest and break down lactose. Well, we don't need to make these lactose enzymes all the time if there's not any lactose around. Right? So there's no lactose to digest. We don't need to waste time and energy making these lactose enzymes. So in the absence of lactose, our repressor will bind to the operator and prevent the transcription from occurring. Right? So we won't produce those lactose enzymes. However, when lactose is present, right, so now there's lactose in the environment that we can utilize for our metabolism, right? Lactose serves as kind of the key to unlock this repressor protein. Right? So when lactose is present, it binds to our repressor protein and inactivates it. So now transcription can occur. RNA polymerase can bind to the operator, and we can synthesize those enzymes for lactose utilization. So in this example, lactose is our inducer. So it's what's going to induce production of these enzymes and turn on this gene. We now know that DNA and genes code for proteins and enzymes that regulate all cellular functions. So any change to that DNA results in genetic variations that can impact any or all aspects of a cell's function and shape. So from things like biofilm formation, pathogenicity, its antibiotic resistance, metabolism, the list goes on and on. So the survival or obliteration of these new genotypes, right, or genetic combinations is called natural selection. So changes in the genetic material, like mutations, which is a permanent change to that base sequence of letters in the DNA, right? So these mutations are really what increase genetic diversity and help drive this process of natural selection and evolution. So this mutation and change can be disadvantageous or even lethal if the cell loses a trait that it needs. Or it could be beneficial with maybe a new or enhanced ability. Or it could be neutral with no real change at all. So mutagens are anything that can generate a mutation. So mutation generating chemicals or substances. Spontaneous mutations occur in the absence of one of these mutagens. So just kind of a random, spontaneous, by chance mutation, which is typically going to involve mistakes during that DNA replication and cell division. So there are several types of possible mutations, but many mutations will be neutral or silent and cause no real change in the activity of a gene. These commonly occur when just one nucleotide is substituted for another in the DNA. But remember, because of that degeneracy of the genetic code, that resulting new codon might still code for the same amino acid. So in this example, if our U right here is mutated to any of the other bases, really, right, we're still going to have the same amino acid and the same protein that has the same structure and function. So even if the amino acid is changed, though, the function of the protein may not change if that amino acid is maybe a non-vital portion of the protein, or if it's chemically similar enough to the original. So a base substitution, sometimes called a point mutation, is our most common type. So it just involves a single base change in the DNA. But these single base changes can have dramatic effects. So this example showing, so during DNA replication, our thymine is incorporated opposite a guanine by mistake. So normally this would be a cytosine. So we accidentally put a thymine here instead. So if our DNA polymerase proofreading doesn't catch this typo in the DNA, in our next round of replication, that adenine is now going to pair with thymine and yield an AT pair. So this would be a correct base pair. But this adenine doesn't know, right, that it was supposed to be a cytosine originally. So now when our mRNA is transcribed from this DNA with our new substitution, a codon is produced that codes for a different amino acid, right? So we're supposed to have two cysteines linked together with a peptide bond, not a cysteine and a tyrosine. So this could change the entire structure and function of our protein. A missense mutation is when the base substitution results in change of an amino acid. So this can have dramatic effects. Sickle cell anemia is caused by just one single letter point mutation in the gene for globin. Right? So hemoglobin in the blood is just a protein. Right? So it has a gene in the DNA that has to be transcribed and translated. Well, this one letter difference mutation changes our amino acid, so it's going to change the structure and function of those blood cells. A nonsense mutation is when that base substitution results in a premature nonsense or stop codon. So this occurs in the middle of a gene, in the middle of the RNA. It can prevent synthesis of a complete protein, so it will be prematurely terminated before it adds all of the required amino acids. Frame shift mutations involve the insertion or deletion of one or more nucleotide pairs. So remember, the genetic code is read 3 bases at a time, right, called codons. So if we just add or remove one letter, right, now we're shifting our reading frame, right, for translation. So if you just delete one pair of nucleotides from the sequence in the middle of a gene, it's going to cause many changes downstream from the site of the original mutation. So it won't just be one incorrect amino acid. Every amino acid coded for after that point will then be affected. Eventually, a nonsense or stop codon will be reached to terminate this inaccurate translation. So in this example of your frame shift mutations, we have our original sequence, the fat cat ate the rat. So if we just delete one letter, we delete the C, now it's the fat atta tet her at, right? So now it doesn't make sense anymore. Our reading frame has shifted. So everything before that mutation is correct, right? But everything after that point mutation downstream from that is going to be affected. Same thing with an insertion. If we just insert an extra letter in the middle of our sequence, the fat car tat eth erat. So shifting that reading frame shift mutations. Spontaneous mutations occur in the absence of any mutagen or mutation generating substance. So we said occasional mistakes made during DNA replication. So we have these built-in proofreading mechanisms. So DNA polymerase that synthesizes new DNA, right? It's an enzyme that makes new DNA polymers. Occasionally can make a typo, right? But is pretty good at noticing its mistakes and correcting them right away. But it's not perfect. Occasionally, errors do slip through the cracks and have these permanent mutations. Chemical mutagens are substances that can generate mutations. So one type of chemical mutagen is nitrous acid. So this causes adenine to bind with cytosine instead of thymine, right? So normally A binds with T, but in the presence of nitrous acid, the adenine is going to incorrectly pair with cytosine. So now when this mutated DNA replicates, that mutation is going to be carried on down the line. Nucleoside analogs incorporates into DNA in place of the normal base, so they cause mistakes in that base pairing. So these analogs are, think, analogous or similar, very chemically structurally similar to our normal bases, just minor modifications, but they're similar enough to be able to incorporate into the DNA and cause those mismatched pairings. Radiation, we know, is also capable of causing mutations. Right? So UV radiation is well known to cause skin cancer. Ionizing radiation, like X-rays and gamma rays, cause the formation of ions within the nucleotide. So they physically break apart the backbone, that sugar phosphate backbone of the DNA molecule. Ultraviolet UV radiation causes covalent bonds between adjacent thymine. So normally the thymines would bind to adenines on the complementary strand, but UV radiation causes them to bind to one another instead. There are enzymes called Photolyases that can work to repair this UV damage, use light energy to break the bonds between these thymines. So they're going to use light. Photo means light. Lyase, so as in lysis, so we're splitting apart, and aces, right? So it's enzyme. So it's an enzyme that uses light to split apart these bonds. Genetic recombination involves the exchange of genes between two different DNA molecules. So this helps to increase genetic diversity, which helps drive evolution. So in this example, we have DNA from two cells that we have aligned. So DNA from the donor will align in complementary base pair with our recipient DNA. So using the aid of enzymes to help catalyze the joining of the two strands, our end result is a chromosome that contains new DNA sequences. Crossing over is another way for genetic recombination in eukaryotes during sexual reproduction. So during meiosis, beginning stage of formation of the sex cells, the gametes, the chromosomes will cross over and exchange some genetic material. So the two chromosomes will break and rejoin, resulting in the insertion of foreign DNA into the chromosomes. So now see, our blue chromosome has some genes from the red, and our red chromosome swaps some genes with the blue. So the end result is that for every round of meiosis, or sex cell production, you get 4 genetically different cells. Vertical gene transfer is the transfer of genes from an organism to its offspring. It's kind of like a family tree. You have your children, the parents, the grandparents. So there is a genetic similarity in inherited chromosomes. Horizontal gene transfer is a transfer of genes between cells within the same generation, right? So maybe neighboring cells that aren't directly related. So vertical gene transfer, parent to daughter cell. Horizontal gene transfer, going from donor to recipient. So these two cells, we're going to exchange DNA. It would be horizontal, right, where this is vertical. So obviously horizontal gene transfer is much more rare than your standard vertical gene transfer. But there are a few ways that microbes can undergo horizontal gene transfer and recombination. So one of those ways is transformation. So in transformation, genes are transferred from one bacterium to another as essentially naked DNA in a solution. So Griffith's experiment in the 1920s showed that genes from dead bacteria could enter living cells and change them genetically so that their offspring became more virulent. So we have a living encapsulated bacteria that's injected into a mouse. So the capsule on this bacteria makes it more pathogenic. So then we grew colonies of the same encapsulated bacteria from the dead mouse. So we have another mouse where we have living non-encapsulated bacteria, and the mouse remains healthy. Because they don't have the capsule, the mouse's immune system is able to fight them off. So a few colonies of the non-encapsulated bacteria were able to be isolated, right, but they were easily destroyed by the phagocytes, right, of the immune system. So in our third scenario, we have some encapsulated bacteria that have been killed via heat. Right? So the cells have been killed, but their genetic material is still in there. Right? So we inject the dead bacteria into the mouse. Right? Mouse remains healthy. No colonies were isolated. Right? These cells are dead. They can't replicate. So the DNA would just degrade by the mouse's immune system. However, if we combine the living non-encapsulated cells with our heat-killed encapsulated bacteria, right, mix them together in the mouse, now the mouse dies, right, and we're able to grow colonies of new encapsulated bacteria. So these dead encapsulated bacteria, right, can't reproduce on their own. So what happened is transformation. So the DNA was released from this dead cell, right? So here we have maybe some capsule DNA, right, in a living cell is essentially just going to pick up this DNA from the environment. Recombination occurs between the two chromosomes, and it now contains the genes for capsule production. So now we have a genetically transformed, recombined cell. So in nature, some bacteria can just release their DNA to the environment after death or lysis. So when the cell membrane disintegrates, the DNA is just now floating freely in the environment. Other bacteria can potentially encounter that DNA, take up those fragments, and incorporate them into their own chromosomes by recombination. Transduction in bacteria is when DNA is transferred from a donor cell to a recipient cell via a bacteriophage or a virus. Right? So instead of the cell picking up random DNA right off the street on its own, we have foreign DNA delivered via a virus vector. So these phages or bacterial viruses use bacterial cell enzymes to synthesize and package their own virus proteins in DNA. So remember, viruses aren't technically living things because they can't reproduce on their own. They have to hack into a living cell's machinery, metabolic machinery, to make new viruses for them. So some of that bacterial DNA occasionally may be packaged inside these new viruses, right, kind of inadvertently. So we have our original donor bacterial cell with the purple chromosome, the dark purple, and a virus, right, a bacteriophage virus injects its viral DNA, shown in pink. So the virus DNA is going to be replicated by the host cell, right? And in the process, its own chromosome is going to be broken into pieces. So occasionally during assembly of these new viruses, instead of packaging virus DNA in our capsids, right, or our virus coats, some of that bacterial DNA may get inadvertently packaged in with the viruses. So now we have another virus, bacteriophage, that can infect another cell, but it's carrying this purple bacterial DNA. So a phage carrying bacterial DNA infects a new host cell, which are recipient cell. So the original bacterial DNA that's been carried by our virus can now recombine and incorporate itself into our new cell. So generalized transduction is just when this occurs randomly, which is the most common type. Specialized transduction, though, is where specific genes are targeted and packaged inside a phage and transferred to the recipient cell. Plasmids and transposons are considered our mobile genetic elements, so they're not part of the primary chromosome. So they're able to move from one chromosome to another or from one cell to another. And because they're not part of that primary chromosome, they don't carry those regulatory structural genes. They usually only carry bonus genes, so things like antibiotic resistance, capsule production. So the plasmid, we said, was our extra chromosomal, your primary chromosome, and then the plasmid. So the plasmids are able to be transferred between cells. So it's a little bit different from transformation because it's going to require cell-to-cell contact. So with transformation, we just had one cell encountering some genetic material. With plasmid conjugation, we have to have two living cells, and they require contact via this sex pilus. So I said plasmids are those self-replicating circular pieces of DNA that often code for those bonus genes, so things that enhance the pathogenicity or virulence of a bacterium. Conjugative plasmids are going to carry the genes for those sex pili and ability to transfer the plasmids. Right? So that conjugation pilus, we have to make this mating bridge, right, or this physical contact for the genetic material to pass from one cell to the next. So this conjugative plasmid or this sex plasmid is sometimes referred to as the F factor. So an F positive cell has, right, that sex pilus ability, the conjugation ability. So it will transfer that plasmid to an F negative cell. Right? So once our recipient receives the new plasmid, it's now considered an F positive cell and can conjugate with other cells and continue passing this plasmid. Dissimilation plasmids encode for enzymes for the catabolism or breakdown of unusual compounds. So this can allow cells to use exotic carbon and energy sources like petroleum for survival. So this would be helpful in things like oil spills. if we can use kind of a bioremedial approach to just have microbes kind of organically break down those oils. So many of these are currently being investigated for possible use in cleanup of environmental waste. Resistant factors, or R factors, are going to code for antibiotic resistance or resistance to toxins and heavy metals. So we have on this plasmid our pili and conjugation proteins, mercury resistance, heavy metal resistance, antibiotic resistance. And we said mutations, and this genetic recombination is what creates cell diversity. And diversity is the raw material for natural selection and evolution. Natural selection acts on populations of organisms to ensure their survival and fit for a particular environment. So, classic example of natural selection is the long-necked giraffe. So, how did giraffes' necks become so long? So, basic principles of natural selection state that each species show variation, right? So there's some differences within a population. Everybody has their own traits. Right? So we have some shorter neck giraffes and some long neck giraffes. Right? So within these species, there's going to be competition for food, mates, right? Habitat. So the better adapted members of these species are more likely to survive and reproduce. So if you're better able to acquire food, right, nutrients, a safe habitat, right, and find a mate, you're going to have more offspring that carry your long neck genes. So the short neck giraffes, right, weren't able to reach the leaves in the trees as well as the long neck giraffes. So over time, several generations, right, the short neck giraffes became less and less common, right, until their short neck genes essentially became extinct. So the survivors are the ones that pass on their better genes to their offspring, who will also have that beneficial trait and be able to better survive. So we see natural selection happening today with things like antibiotic resistance. So the way antibiotic resistance happens is you start with a non-resistant bacteria. Right? Bacteria are going to multiply by the billions. Right? Bacteria grow exponentially. Right? Occasionally, mutations will occur. And then occasionally, some of those mutations will make that bacteria resistant to antibiotics. So in the presence of that particular drug, only the ones with that resistance mutation survive. So they're left to repopulate the colony. So now all of the bacteria in this infection are resistant to our drug treatments. So theory of evolution in a nutshell, right? So summing up Darwin's 4 postulates for natural selection. Right. So number 1 states that there's variability within a population, right? So genome mutations are constantly occurring, right? So there is genetic variation. We're not all genetically identical. We do have different versions or alleles of the same genes. So some of these variations are heritable or passed down to the offspring, right? Or these mutations, right, will be passed down to the next generation of cells. So some individuals will be more successful at survival and reproduction than others. So individuals with those particular mutations may survive and reproduce more, right? And therefore, over time, the population as a whole will evolve, right? So there'll be a genetic shift in the population. So in this kind of rudimentary example, so through natural selection, the thumb developed proved beneficial, right? So having just 1 finger or 3 or 4 fingers, right, wasn't as useful as having, right, 5 fingers. So over time, this process or buildup of mutations can result in populations that specialize for particular ecological niches or habitats, and can eventually result in emergence of new species. Okay, so basically saying that survival and reproduction is not entirely random. It is linked back to some of these individual variations in traits. So remember we said that a lot of the mutations are just mistakes in copying. So any mutation is a change in the DNA that ought not be there, right? So we shouldn't want to change our genetic sequence, right? But if you think about it, evolution is driven by these mistakes, right? These mutations. So all of life and evolution and diversity that we have today is the result of a million or more mistakes that have kind of snowballed into life as we know it today.